Professor Hawley. My name is Javier Aleman and I'm working with Marcos Dominguez, and together we are going to explain our organelle, which is the nucleus. The nucleus is the biggest organelle in the eukaryotic cell. It contains the cell chromosomes. The pores in the nuclear membrane allows the passage of molecules in and out of the nucleus. The nucleus is an organelle found in eukaryotic cells, as I previously said, and inside is fully enclosed nuclear membrane. It contains the majority of the cell genetic material. The material is organized as DNA molecules, along with a variety of proteins, to form chromosomes. The nucleus controls and regulates activities of the cell, growth and metabolism, and carries the genes, a structure that contains the heredity information. In this picture, we can see the anatomy of the nucleus. Inside we can watch the nucleolus, chromatin, the nucleus involved. Here are the pores that make the nucleus permeable, the ribosomes, and the endoplasmatic reticulus important. The nucleus contains nearly all the cell's DNA, surrounded by a network of fibrous intermediate filaments and involved in a double membrane called the nuclear envelope. The nuclear envelope separates the fluids inside the nucleus, called the nucleoplasm, from the rest of the cells. The size of the nucleus depends on the size of the cell, of course, and it is contained with a nucleus typically occupying about the 80% of the total cell volume. The nucleus is the largest organelle in animal cells, and in mammalian cells the average diameter of the nucleus is approximately 6 micrometers.
So the function of the nucleus is that they control of the DNA. They are the one that control the genetic information of the cell and thus the hereditary characteristics of an organism. Also, they control the proteins and enzyme synthesis, the cell division and cell growth, the storage of DNA, RNA, and ribosomes, and they regulate the transcription of mRNA to proteins also. So basically the structure of the nucleus. So one of the parts of the nucleus is the nuclear envelope. As the name suggests, the nuclear envelope surrounds the nucleus, separating from the cell cytoplasm. It is a double membrane, and each membrane is a phospholipid bilayer associated with proteins, and the two members are divided by 20 to 40 nanometers of space. These two members of the nuclear envelope are often referred to as the inner and outer nuclear members. The outer membranes continue with the cell's endoplasmatic reticulum, and therefore the space between the inner and outer nuclear membranes looks to the lumen of the endoplasmatic reticulum. We also have the nucleoplasm, which is similar to the cytoplasm of the cell, but is a semi-liquid and fills the empty space inside the nucleus. It is from a protoplasm and surrounds the chromosomes and nucleoli inside the nucleus. It is also various proteins and enzymes dissolved within. The nuclear bodies can we find in the nucleoplasm, and these include structures such as KL bodies, Gemini bodies, and polycomb bodies. KL bodies are between 0.1 to 1 in diameter and can be found in prophylline states, such as embryonic cancer cells. We also have the nuclear lamina, which is in animal cells. Two networks of intermediate filaments provide the nucleus with mechanical support. The nuclear lamina forms an organized meshwork of the internal face of the envelope, with less organized support is provided of the cytosolic phase of the envelope. Both systems provide structural support for the nuclear envelope and anchoring chromosome for nuclear pores. We also have the nucleolus, which is the largest of the discrete densely stained membranous structure known as the nuclear body found in the nucleus. It forms around tandem repeats of DNA coding for ribosomes, RNA, and in this region are called nuclear organizer regions. The main role of the nucleolus are to synthesize RNA and assemble ribosomes. The structural cohesion of the nucleolus depends on its activity, as ribosomal assembly in the nucleolus result in the transient association of nuclear components, facilitating further ribosomal assembly and hence further association. This model is supported by observations that inactivation of rDNA is resulting in intermingling of nuclear structures. And finally, we have the ribosomes, which are ribosomes are made of proteins and ribonucleic acid, abbreviated as RNA. In almost equal amounts, it comprises of two sections known as subunits. The thinner subunit is the place that the mRNA binds and it decodes, whereas there's a big subunit in the place the amino acids are included. The ribosomes are organelles located inside the animal, human cell, and plant cell that are situated in the cytosol, bound and free floating to the membrane of this coarse endoplasmatic reticulum. Their main function, when it comes to is that they assume the role of bringing together amino acids to form particular proteins, which are important for completing the cell's activities. Another important thing that we can find inside the nucleus and that actually compose the DNA is the chromosome. The cell nucleus contains the majority of the cell genetic material in the form of multiple linear DNA molecules organized into structure called chromosome. Each human cell contains roughly 2 meters of DNA. During most of the cell cycle, these are organized in a DNA protein complex known as chromatin. Under cell division, the chromatin can be seen from the well-defined chromosome family from a karyotype. A small fraction of the cell genes are located instead in the mitochondria. Chromosomes are not visible in the cell's nucleus, not even under a microscope when the cell is not dividing. However, the DNA that makes up chromosome becomes more tightly packed during cell division and is then visible under a microscope. More of what researchers know about chromosome was learned by observing chromosomes during cell division. Each chromosome has a constriction point called the centromere, which divides the chromosome into sections or arms. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes.
We can think about the nucleus as the brain of the cell because it controls most of the functions. Without the nucleus, the cell will not work. There will be no cell division. Protein synthesis will either cease or incorrect proteins will be formed. All this will result in cell death. But what disease could be caused by malfunctions in the DNA, in the nucleus or nucleolus? For example, defective nucleoli have been implicated in several hereditary diseases. Most recently, also in neurodegenerative disorders such as Alzheimer or Huntington disease. For example, this is how a brain with Alzheimer looks like, and this is how a brain with Huntington disease looks like. It's pretty similar. Mutation in some of the protein that make ribosome cause disorder characterized by bone marrow failure and anemia early in life, followed by elevated cancer risks in middle age. However, the available ribosomes are defective and cause change in gene expression pattern that can result in cancer. But what would happen if the flagella stopped working? For example, secretion would not be possible, so a buildup of material would occupy other organelles in the cell. Cell will not be able to move, and the flagella, the cell will no longer be able to move. Another example is, it is known that each person needs an exact number of chromosomes. We all know about Down syndrome. Down syndrome is a genetic disorder caused by an abnormal cell division result in extra genetic material in the chromosome 21. Down syndrome causes distinctional facial appearance, intellectual disability, and delays in the knowledge.
Well, Professor, this is our video. I hope you all enjoyed all the information we share with you and with the class. Once again, it's been a pleasure. Javier Alemán and Marco Dominguez.
Thank you very much.